Hello, students and my dear friends. Myself, Madhu Vanja, Assistant Professor of Microbiology. In this session, we'll discuss about history and introduction of microbiology. This is very interesting topic. So, where you learn how the microbiology science has started or it is evolved, and like how the Anthony van Leeuwenhoek, Louis Pasteur, Robert Koch, and other scientists like how they contributed for the development of the like microbiology. So, what are their discoveries? We'll discuss in this session. If you see the definition, so as you know, so microbiology it is the study of organisms which are usually invisible to the naked eye, which means you cannot see with your eyes. So to visualize the like microorganisms, you need an equipment called microscope. So with the help of the microscope only, you can see the like microorganisms. So we cannot see them with the like naked eyes. So they are microscopic structures. So if you see the word microbiology, so in the Greek, the micro means small and bios means life and logy means study. So which means it is the study of small life forms. And if you see the term microbiology, it was coined by the scientist Louis Pasteur. So Louis Pasteur is considered as father or founder of modern microbiology. So, medical microbiology. So, in the medical microbiology, what we discuss, we deal with the causative agent of the disease or infection, diagnosis, prevention, and treatment. Which means, so what are the organisms? Like, what are the bacteria causing infections? So, what are the viruses they are causing infections? So, what are the fungi causing infections? So, how they are entering into our body? So, how what are the toxins they are producing, and what are the enzymes they are producing? And how that toxins and enzymes contribute for the like infection or disease. And diagnosis part will discuss, such as like what are the samples to be collected, like how to process the samples, so what are the different laboratory diagnostic methods. And prevention, such as what type of vaccines are available to prevent that particular infection. And we'll discuss even treatment strategies. So if you see, this is the development, like historical development of the microbiology. So in the first century BC, scientists Varro and Colmella, so they gave the concept of Animalia minuta. So what is this Animalia minuta? What is the concept of Animalia minuta? So according to this concept, the diseases are caused by the invisible organisms. So according to the concept of Animalia minuta, the diseases are caused by the invisible organisms. Next, in the year 1546, Castorius is from Verona. So he gave the concept of contagion is the cause of the syphilis. So what is mean by syphilis? So syphilis, it is a STI, sexually transmitted infection, which is caused by bacteria called Treponema pallidum. So he told that contagium vivum. So contagium vivum is the like fluid or living germs. So they are the like a cause of the like infectious diseases. Okay. So he told that the infectious diseases are caused by the tiny living particles, which is called as the seminaria or contagium vivum. Next, in the year 1590, so Jensen, like he gave the like a hand lens. So what is the hand lens? It is a like hand magnifier. Like so, it is a type of like microscope only. So which is a like magnifier. So this is mostly used in the like microbiology laboratory to examine the like colonies. So next, in the year 1683, scientist Antony van Leeuwenhoek, so he discovered the like first microscope and he gave the concept of animal culture. So regarding the Antony van Leeuwenhoek and his discoveries, we'll discuss in detail in the next slide. Next, in the year 1678, Robert Hooke. So Robert Hooke, he was a physicist and a microscopist. So he prepared the first compound microscope. Next, in the year 1745, John Needham, he is an Irish priest. So he gave the concept of abiogenesis. So he proposed the like a theory of abiogenesis. So theory of abiogenesis, it is also called as theory of spontaneous generation. So according to this theory of spontaneous generation, So all the organisms they raise spontaneously from the non-living matter. Okay, so life that can arise spontaneously from the like non-living matter. So even though mm, it is wrong, like theory of abiogenesis or theory of spontaneous generation is wrong, so later it was disproved by the scientist Louis Pasteur. But in the olden days most of the people they believed the like a theory of spontaneous generation. So here sometimes like they observe such as some of the flies or maggots or small frogs, they are coming out from the like a decomposing material. So they believed that they are directly coming out from the like a decomposing material, but that is not come, that is not true. So now we are not teaching the like a theory of abiogenesis, so we are teaching the like a theory of biogenesis, which means always life will come from the life only. Next, in the year 1836, Schach and Swan they proved that air contained the like microscope microbes. So they are the supporters of the like biogenesis theory of biogenesis, which means life will come from the life only. Next, in the year 1840, Oliver Holmes is a poet and physician. So he gave the concept of contagiousness and puerperal fever. So that is a infection after the like delivery in the like mothers. And in the year 1846, Ignis Semmelweis, so he like gave the concept of like cause, concept, and prophylaxis of childbed fever. So childbed fever is nothing but the like puerperal fever only. So here what is this contagiousness and what is this puerperal fever? So puerperal fever, as I told you, it is also called as the like a childbed fever. So it is spread by the physicians and nurses who attended the childbirth after handling the infected patients. So in the olden days, so most of the like clinicians, they are they didn't practice like a hand washing. So they used to treat the like patients so without washing the like hands. So that's what like the infection has spread from like one woman to the like another woman. So here Ignis Semmelweis, so he insisted on the like cleanliness and disinfection of hands, cloths, and instruments before attending the like deliveries. So Ignis Semmelweis, so he was a doctor. He worked in the like Vienna General Hospitals. So he observed that like the mothers who are delivered by the like doctors and medical students, they have higher mortality rate because of the like puerperal fever than the women who are delivered by the like midwives. So he traced he identified that, so like the cadaveric particles so which are on the like doctor hands, they are causing this infection. So he introduced hand washing with the chlorinated lime solution before examining the patients. So that's what it reduced the mortality rate. So he was the Ignis Semmelweis was the first person who stressed the importance of the like hand washing. So if you wash your hands properly, you can prevent almost 80% of the infections. Next, in the year 1835, Agostino Bassi, so he told that like a muscardine disease, that is a silkworm, like a disease is due to a fungus. So here, what is this muscardine disease? So there will be like a white powdery spots on the like silkworms. So like so it is like there is like massive like loss of the like silkworms. So that is a loss for the like silk industry. So he proved that it is because of the like fungus. Next, we'll discuss about Antony van Leeuwenhoek. So Antony van Leeuwenhoek, he born in the year 1632 and died in the year 1723. So he was a draper from the Delft city of the Holland. So he was a cloth merchant. So by profession, he was not a formally trained scientist, but like he has like hobby of the like grinding the like lenses. So he has a fashion like on the grinding of the like lenses, and he used to prepare the like new lenses. So here with his lenses, generally he used to observe the like quality of the cloth. He used to check the like quality of the like cloth. So during that process, like he got interest towards the nature. He started examining the like various materials from the like nature. So he took like his tooth scrapings and he examined under his like microscope. So he was the first person who prepared the like microscope. So he observed the like living cells with the like with his simple microscope. So he ground his like own lenses, and like he took the like tooth scrapings and he mixed it with the water and he observed under his microscope. So he named them as like animalcules. So actually, they are the various forms of the like bacteria as well as the like protozoan what we know today. So they are the like rod-shaped bacteria, spiral-shaped bacteria, and he identified the like various protozoans. So, but by the time of the Anthony van Leeuwenhoek, no one knows about the like existence of microorganisms. So that's what he named them as like animalcules, which means like small animals. So why he named them as like animalcules? Because some of them are like showing the motility movement. So that's what he named them as like animalcules. So in his lifetime, he prepared more than 400 simple microscopes by the hand, and he wrote almost 300 letters to the like Royal Society of London. So Royal Society of London, it is the oldest national scientific institution. So he communicated his findings, like means he wrote the like letters to the like Royal Society of London by describing his like findings. But the significance of these observations was not then realized. So no one recognized Anthony van Leeuwenhoek work because Anthony van Leeuwenhoek, he has no formal scientific education, or he was not a trained scientist. And one more thing is like he wrote his letters in the like Dutch, but not in the like Latin. So Latin was like a scientific language. And next, he was the first person who prepared the like microscope, and he was the only person who knows how to prepare the like microscope. So his microscopes were unique and secret, and he didn't share the formula like how to prepare the like microscope with anyone. So that's what they thought that they're all like imaginaries only. So if you see in this picture, so this was the like first microscope which was prepared by the like Anthony van Leeuwenhoek. And in this picture you can see, so these are the like various forms of the like bacteria. So like rod shaped bacteria, like spiral shaped bacteria. So he named them as like animalcules, but actually they are the various forms of the bacteria. Next, Louis Pasteur. So Louis Pasteur, like he was a French chemist. He is considered as father or founder of the modern microbiology. So he told that fermentation is a microbiological process. So what is mean by fermentation? Fermentation, it is the breakdown of carbohydrates which results in the production of alcohols, acids or gases in the absence of the oxygen, which means it is the anaerobic breakdown of the carbohydrates by the microorganisms. Generally bacteria or yeast, they break down the like sugars such as carbohydrates, which results in the production of alcohols, acids and gases. And he told that uh, so actually in the olden days, like the government of France has faced a problem that wine was becoming sour in the taste. So they thought that it is maybe due to the like chemical changes. They thought that it was a chemical process. So they asked like Louis Pasteur to investigate that, and he identified some of the like yeast cells in that one, and he told that microorganisms are responsible for this fermentation. And he gave the like a solution by heating the liquids at a low temperature uh, for around like a few minutes, so which reduces the like which kills the like harmful microbes. So he told that different types of fermentations are caused by the different kinds of the like microorganisms. So there are different fermentations are there, such as alcoholic fermentation, lactic acid fermentation, like that. And next, uh, he gave the like a pasteurization of milk. So what is mean by pasteurization? So pasteurization, it is the process of killing of microbes which are present in the milk or otherwise other food material. So by heating them at low temperature, that is 60 to 70 degrees centigrade for few minutes, and we rapidly cool to the like lower temperatures. So this will kill the like microorganisms which are present in the like milk, and still we are using this pasteurization technique. Next, he introduced methods and techniques of cultivation of the bacteria, and he proposed the germ theory of the disease. So what is mean by germ theory of the disease? So according to the germ theory of the disease, the infections or diseases are caused by the germs, such as nothing but microorganisms. Next, he disproved the theory of spontaneous generation. So what is mean by theory of spontaneous generation? As we discussed, theory of abiogenesis. So here, he disproved the theory of spontaneous generation by using of his swan neck experiment. Like next, he was the first person who used liquid media to grow the microorganisms, such as he used nutrient broth to grow the microorganisms, and he introduced sterilization. So what is meant by sterilization? It is a process of killing of microorganisms. So he developed the steam sterilizer, autoclave, and hot air oven. They are all the equipments which we use in the sterilization. And he studied it about the silkworm disease, anthrax, chicken cholera, and hydrophobia. So like he was the he studied it about the silkworm disease, and he told that the silkworm diseases are due to the febrile, which is caused by the protozoan, and flecherae, which is caused by the bacteria. And he studied it about anthrax. So anthrax is caused by the bacillus anthracis bacteria. So generally it infected the sheep as well as the cattle. And he worked with the chicken cholera bacillus, and he had he identified the concept of the attenuation. So here he introduced the live attenuated vaccines. So here if you see, so actually Louis Pasteur, he was working with the a chicken cholera bacillus. So here during the a vacation, so he left his old cultures like that only, and once he come back from the vacation, he used the same aged culture of a chicken cholera a bacteria. He injected into the a healthy chicken, and surprisingly this healthy chicken is free from the cholera. It didn't develop any disease, so it has mild symptom and it recovered. And after few days, again he took the a fresh culture of the uh, uh, cholera bacterium, and he injected with the aged culture. So here this a bird, it has already injected with the aged culture, and to this bird again he injected the a fresh culture broth, and this chicken, it didn't develop any diseases, so it is free from the cholera. And again he took the same fresh culture of the bacteria, and he injected into the normal healthy chicken, and it died due to the cholera. So here what happened here? So as this a culture media left for longer time, so this bacteria got attenuation, which means it lost its virulence, which means it do not have ability to produce the disease, but it retain the antigenicity. So this is laid for the foundation for the development of the vaccines. Next, like he was the first person who introduced the anti-rabies vaccine. So actually, Louis Pasteur, while he was working for with the hydrophobia. So what is mean by hydrophobia? That is fear of water. So that is rabies. So rabies, it was a fatal viral disease which is transmitted by the animal bite. So especially by the bite of the dog. So while he was working with the uh, hydrophobia, which means rabies virus, he tried to isolate the a causative agent of the rabies, but he could not. He thought that it is due to the bacteria, but it is not caused by the a bacteria, so he could not isolate this one. So if you, in case of rabies, so if any person is affected by the dog bite, so if they develop the a symptom, sure they will die. So there is no treatment for the rabies, and uh, like so he prepared the anti-rabies vaccine. So in this picture you can see. So he took the nervous tissues, uh, like uh, he took the saliva from the infected dog, and he injected into the rabbit body, and this virus has this virus has grown in this rabbit body, and he took the like a nervous tissue, which means the spinal cord of the like rabbits, and he dried this spinal cords in the like sterile air for few days. So this sterile air reduced the like virulence of the like organism, and he prepared the like a series of the like spinal cord suspension, which gradual decreasing of the like virulence, and he gave uh, into the like uh, uh, Joseph Mister. Okay, so Joseph Mister, he was like a 9 years old boy. He was affected by the like a dog bite, and almost he has the like 14 wounds. So here he took this vaccine and he gave the like a 13 injections in 10 days. And surprisingly, this boy such as Joseph Mister survived from the like rabies, and it laid a foundation for the development of the like vaccine, which was the greatest achievement in the field of the like microbiology. Then Pasteur became world famous, and people from all the countries came to get the like vaccine. And in the year 1888, so people only contributed funds to the like Louis Pasteur, and they built the like Pasteur Institute of the Paris. Next, Joseph Lister. So Joseph Lister, he was the professor of surgery at Glasgow Royal Infirmary. So he applied the like Pasteur work, and he introduced antiseptic surgery in the year 1867. So here in the olden days, so most of the people, they died after the like surgery. It was not because of the like surgery, it was because of the infections after the like surgery. So as we discussed, so there were like no proper sterilization method or disinfection methods in the olden days. So that's what microorganisms they causes the like infection after the like surgery. So here like Joseph Lister, he used carbolic acid. So carbolic acid is a phenolic component. So he sprayed carbolic acid as a spray during the like surgery. So this carbolic has like it spreads in the like environment, and it killed the like microorganisms which are present in the like environment, so that the microorganisms could not reach to the surgical site and the infections are prevented. And not only that one, he used carbolic acid so for hand washing, and he disinfected the like cloths and other materials of the like means surgery equipments by using of the like a carbolic acid, so which killed the like microorganisms. So he is considered as father of antiseptic surgery. Next, Robert Koch. He was a German general practitioner. He is considered as father of bacteriology and father of medical microbiology, because most of the medically important bacteria was discovered by the scientist Robert Koch, and he perfected the bacteriological techniques. So actually, Louis Pasteur, he gave the like techniques for cultivation of the like bacteria. So Robert Koch perfected that techniques, and he isolated pure cultures of the bacteria for the first time. So what is mean by pure culture? So pure culture means culture consists of single type of microorganism, and he discovered anthrax bacilli, which means Bacillus anthracis, which is the causative agent of anthrax. So it is disease of the like sheep, cattle, and other animals. It is a zoonotic infection. And he discovered Vibrio cholera vibrio in the year 1883, so which causes cholera. And he discovered Mycobacterium tuberculosis, which is the causative agent of the like TB, like pulmonary tuberculosis, in the year 1882. And he described about the hypersensitivity reaction. So what is mean by hypersensitivity? Such as allergy reaction. He introduced staining techniques. So what is mean by staining? So staining means applying of stains or dyes or colors to the like microbial cells. So to visualize the like microorganisms, to visualize the like bacteria, so we have to apply the like stains to the like bacteria. So these techniques was given by the scientist Robert Koch, and he was the first person who discovered and used solid medium in the like bacteriology. So in the beginning of the microbiology, there were only like liquid culture medias, and even like there were some solid culture medias are there, such as like cooked cut potato medium and other culture mediums. So in the initial days, they used like a gelatin for preparation of the like a solid medium, but it was not satisfactory because gelatin liquefies at the like a room temperature. So at like 20 to 25 degrees centigrade, the gelatin liquefies. So they were facing lot of problem with this like a solid culture medium. So one day, Robert Koch's like laboratory assistant, her name was like Fanny Hussey. So she suggested the usage of the like a agar agar. So agar agar, so it is extracted from the like a red algae, that is from Geladium species. So has like a properties such as it has solidifying property. So it has no color, like no odor, no taste, nothing. So just it acts as like a solidifying agent. So he was the first person who used to agar agar for preparation of solid culture medium, and he was the first person who used laboratory animals for the like experiments, and he described hanging drop method. So what is hanging drop method? Hanging drop method is to observe the like motility of the like bacteria. Next, very important thing is like Koch's postulate. So as we discussed, like Louis Pasteur proposed the germ theory of the disease. So what is meant by germ theory of the disease? So according to the germ theory of the disease, the infections are caused by the germs or microorganisms. So Louis Pasteur proposed it, and Robert Koch proved it by his postulate. Okay. So what are those postulates? Like association, isolation, inoculation, and re-isolation. So here, according to the Koch postulates, so if any microorganism follow the Koch postulates, then only it is considered as like a pathogenic organism. Suppose if organism does not follow the like Koch postulates, it is considered as like a non-pathogenic organism. So here, what is association? So here the organism should be constantly associated with the lesion of the disease. So as we are telling, like microorganisms are responsible for the like infections, or like germs are responsible for the like infection. So here always the organism should be constantly associated with the lesion of the disease. This is called as like a association. Then next, isolation. So what is mean by isolation? Isolation means like separation. So when we collect the like a sample from the lesion of this disease, so it should be possible to isolate the organism in the pure culture. So when we took the like a sample from this lesion, so we should able to isolate the organism in the pure culture. So what is meant by pure culture? Pure culture means culture consists of the single type of organism. Then next, inoculation. So what is mean by inoculation? Inoculation means to introduce. So here the isolated organism in the pure culture, when it is inoculated into the suitable laboratory animal, then the laboratory animal also should produce like similar disease. So whatever the type of lesions here we have observed in the association, so same type of lesions we should see in the like animal. Next, re-isolation. So what is meant by re-isolation? So again, it should be possible to re-isolate the same organism in the pure culture from the lesion which is produced in the like experimental animal. So if you see in the like third step, so here the laboratory animal produced the like disease. Suppose if you collect the like sample from this laboratory animal, so we should isolate the like same organism which we have isolated earlier. Okay, so these are the like Koch postulates. So in this picture you can see, so you can understand clearly. So according to the association, so always the microorganism should present. So yeah, in the lesion. So in every case of the like a disease. So isolation, which means when we collect the like a sample from this infected like a animal, so we should isolate the like a organism in the like a pure culture form. And here the organism is isolated and it is inoculated into the like laboratory animal, and the laboratory animal also should produce like a similar type of disease. And again, when we collect the like a sample from this laboratory animal, so we should isolate the like a bacteria, so which we have isolated earlier. So here you can see the like a same anthrax bacilli. So here Robert Koch, he proved the like a Koch postulates by using of the Bacillus anthracis bacteria. And later, one more additional criterion also added, that specific antibodies should be demonstrable in the serum of the like a patient. So here, as this microorganism is acting as antigen, so against this antigen, antibodies are produced. So we should able to identify the specific antibodies in the serum of the patient. So here, if organism follow the like a Koch postulates, then only it is considered as the like a pathogenic organism, or otherwise it is considered as non-pathogenic organism. But there are some exceptions to the like a Koch postulates. Suppose if you see like Mycobacterium leprae, Treponema pallidum, and Neisseria gonorrhoeae. So even they do not follow the like a Koch postulates, so still they are considered as like a pathogenic organisms. Next, Koch's phenomenon. So this is like hypersensitivity reaction. So when guinea pigs, which are already infected with the like tubercle bacilli, the guinea pigs which are already infected by the tubercle bacilli, so they show the exaggregated inflammatory response when injected with the tubercle bacilli or protein. So again, when we give the like tuberculin injection or protein, they show the exaggregated inflammatory response, which means hypersensitivity reaction. So that is called as like a Koch's phenomenon. Next, Paul Ehrlich. He was a German scientist, and he was one of the like student of like Robert Koch, or like assistant of the like Robert Koch. He is considered as father of chemotherapy. He applied stains to the cells and tissues to study their function. So Robert Koch applied the like stains to the like microbial cells. Paul Ehrlich applied stains to the cells and tissues to study their function, and he reported the acid-fast nature of tubercle bacilli. So here Mycobacterium tuberculosis, it is also called as like a acid-fast bacteria. So it has acid-fast nature. So why it has acid-fast nature? Generally, all bacterial cell wall is made up of peptidoglycan, but Mycobacterium tuberculosis cell wall along with the peptidoglycan, they have mycolic acid. So mycolic acid is a wax-like substance. So because of that one, they have the like acid-fast nature. And he proposed the like side chain theory of antibody protection. So what is this side chain theory? So cells have the side chains, which means receptors. So they binds to the like antigens, which are nothing but like what bind to the like antigens? Antibodies. And he discovered the salversan, so which is also called as the magic bullet, which has ability to destroy the spirochete of the like syphilis. So it is a arsenic compound, so which is a first chemotherapeutic agent. So the salvarsan, it has like ability to destroy the Treponema pallidum, which is a causative agent of the like syphilis. And later on, he discovered the like neosalvarsan, and he introduced methods of standardizing the toxins and antitoxins. So he introduced the like methods of standardizing the like dosages of the toxins. Next, these are other important discoveries by other scientists. If you see leprabacillus, which is Mycobacterium leprae, it was discovered by the scientist Hansen in the year 1874. So it is also called as a Hansen's bacillus, and the disease is also called as a Hansen's disease. Next, gonococcus. Gonococcus was discovered by the scientist Neisser in the year 1881, which is what is the scientific name of the gonococcus? Neisseria gonorrhoeae or Neisseria gonorrhoeae. Next, diphtheria bacillus, that is Corynebacterium diphtheriae. It was discovered by the scientist Klebs in the year 1883, Loeffler's in the year 1884. So it is also called as the Klebs-Loeffler's bacillus. Next, pneumococcus, such as Streptococcus pneumoniae. It was discovered by the scientist Franke in the year 1886. And a diphtheria toxin, such as Corynebacterium diphtheria produces diphtheria toxin. So this toxin was discovered by the scientist Rox and Yersin in the year 1888. Next to tetanus bacillus, such as Clostridium tetani. It was discovered by the scientist Kitsato in the year 1889. And plague bacillus, which is a causative agent of plague, it was discovered by the scientist Yersin in the year 1890. And next, Hans Christian Gram in the year 1884. So he discovered the like gram staining. So what is gram staining? Gram staining is the like a basic staining technique in the microbiology. So based upon the gram staining, the bacteria, they are divided into gram positive bacteria and gram negative bacteria. Gram positive bacteria appears purple or violet in the color, and gram negative bacteria appears pink or red in the color. And next, Ivanovsky, in the year 1892, he reproduced mosaic disease in the like a tobacco plant. So here, by the time of the like Louis Pasteur, or by the time of the like Robert Koch, most of the medically important bacteria were discovered. But still, there was some mystery in case of some infections. So even even Louis Pasteur also tried to isolate the like a causative agent of rabies, but he could not. So here, Ivanovsky, he took the infected leaves of the like tobacco plant, so which is the like tobacco mosaic disease. So he took the like infected leaves of the like tobacco plant, and he macerated with the help of the mortar and pestle, and he extracted that juice or sap, that filter, and he passed that like a juice through the like a bacterial filters. So bacterial filters have the pore size smaller than the bacteria, which means the bacteria could not pass through this bacterial filters. Okay, and he passed the juice through the like bacterial filters, and he extracted that juice, and again he applied it on the like healthy leaves, and healthy leaves surprisingly they developed the like a disease, and he gave the conclusion that there are some organisms are there which are smaller than the bacteria. So they are responsible for this tobacco mosaic disease. And later, they were confirmed as viruses by the scientist Besrink. Okay. Next, Loeffler and Frost in the year 1898. So they told that foot and mouth disease agent also can pass through the like bacterial filters. Like how the Ivanovsky did the like experiment, by the same way they did the like experiment. And next to Walter Reed in the year 1902, he proved that mosquitoes carry the like yellow fever agent. So yellow fever, uh, it is caused by the like viruses. Okay, so this mosquitoes carries this like a yellow fever virus. Landsteiner and Popper in the year 1909, they told that poliomyelitis disease is due to the like filterable virus such as poliovirus. Next, good posture in the year 1930. So he gave the like a concept like virus cultivation in the like chick embryos. So as you know, viruses cannot be grown in artificial culture media. So they can be grown in the like chick embryos. Like they require the like living cells. Next, Raska in the year 1934, he introduced the like electron microscope. So generally, the larger viruses like smallpox and some other larger viruses can be seen with the compound microscope, but other viruses cannot be seen with the like normal microscope or compound microscope. So to visualize the like viruses, we need electron microscope. Next, Edward in the year 1915 and D. Herrele in the year 1917, they discovered the like a bacteriophage. So what is bacteriophage? So the viruses which infect the bacteria, they are called as like a bacteriophage. Next, Edward Jenner in the year 1796. So he gave the first scientific smallpox vaccination. So there is an interesting story behind this one. Smallpox, like it was one of the like deadliest disease in the like olden days. Here he observed that the milkmaids, okay, are the women who collect the like milk, the milkmaids who has recovered from the cowpox infection, they are not getting the like a smallpox infection. So cowpox is the like a mild disease which is transmitted from cows to the like humans. So he observed that the women who have recovered from the like a cowpox infection, they are not getting the like a smallpox infection. So he want to prove it. Then he took the scrapings from the like infected lesion from the cowpox. So he took the like infected scrapings from the like cowpox lesion, and he injected into his gardener's son. So this boy name was like James Phipps. He was 8 years old, and he intentionally introduced into the like James Phipps body. And after few days, James Phipps developed the like a cowpox infection. He suffered from cowpox for almost 8 to 10 days, and he recovered fully. And after few days or after few weeks, again he took the like infected material from the smallpox lesion. So the persons who are suffering from the like smallpox persons, so from that like he took the like material, that fluid or pus like material he collected, and he injected into the like James Phipps body, and surprisingly James Phipps, he didn't develop any infection. So this was a greatest milestone in the development of the like vaccines. Okay, so here James Phipps, he could not develop any disease, and it laid foundation for the development of the like vaccines. So here in the Latin, vaxa means cow. So as this material was in first taken from the like lesions, means especially from the like cow material. So that process is called as like a vaccination, which means giving the like a cow material. Okay, so here um, the smallpox, it has been eradicated because of successful and effective vaccination, which is given by the scientist Edward Jenner. Next, Van Behring and Kitsato in the year 1890. So they produced antibody, like means by giving of foreign serum, which is injecting sublethal doses of tetanus toxin into the like a guinea pig. Next, Elham Metnikov in the year 1883, he proposed the like phagocytosis. So what is meant by phagocytosis? That is engulfment of bacteria by the like WBC. Next, Jerne in the year 1955. So he proposed a natural selection theory of antibody synthesis. Bonnet in the year 1957 modified this into the like clonal selection theory. Next, Bonnet, he proposed the concept of immunological surveillance. And next, Sir Ronald Ross, he discovered the like malarial parasites in the mosquitoes. And next, Carrie B. Mullis, he discovered the PCR, polymerase chain reaction. For this contribution, he was awarded with the Nobel Prize in the year 1993. Next, another important discovery is by scientist Sir Alexander Fleming. So he discovered the penicillin antibiotic, first antibiotic penicillin in the year 1928. So actually he was working with the like Staphylococcus aureus bacteria. So once his cultures got contaminated with a like mold, such as a fungi, and he observed that so wherever the growth of the like mold, around that there was no growth of the like bacteria. So there was no growth of the like Staphylococcus. And finally, he extracted that substance and purified, and it is like nothing but the like a penicillin. It was the first antibiotic which has saved millions of people during World War II. So this is the end of the session. If you see the like summary, the microorganisms are living structures of microscopical size. So microscopy can done, which means microscopy is needed to observe the like organisms, microorganisms. Antony van Leeuwenhoek, he was the first person who observed the microorganisms, and he named them as animalcules. And Louis Pasteur developed the techniques of sterilization, and he postulated the germ theory of the disease. Louis Pasteur disproved theory of spontaneous generation. Robert Koch proved germ theory of the disease by his postulates. Joseph Lister used carbolic acid as a spray to reduce the like a post-operative infection. Paul Ehrlich discovered salvarsan. Thank you.